Hello, everyone. Thank you for having me here today. My name is Joaquín Castilla, and I'm calling from bioGUNE in Spain. I'm really honored to share with you some very exciting results from our research that was made possible by your generous support through the CJD Foundation. Let me start with the challenge we are facing. As everyone in this room knows, prion diseases like CJD, fatal familial insomnia, and GSS are devastating and fatal conditions. Currently, there are no treatments available to stop or slow this disease progression. Our urgent need is to develop new treatments that can actually block prion propagation, that's the spreading process in the brain. Our goal is ambitious but clear. We want to develop a therapy that can slow down or even halt this disease process by stopping prions from spreading. So how are we tackling this challenge? We are using an approach called gene therapy with something called dominant negative prion proteins. Now, let me break this down in simple terms. Gene therapy uses modified viruses, but don't worry, they are completely shaped, called AAV vectors, to deliver therapeutic genes directly to brain cells. Think of these as tiny delivery tracks that can cross the blood-brain barrier and reach the affected neurons. The dominant negative proteins we are delivering have three key characteristics. First, they don't misfold spontaneously. They remain stable. Second, they maintain normal cellular function with no toxicity to neurons. And third, this is the crucial part, they block prion propagation by interfering with the misfolding of the endogenous peer proteins. So essentially, our AAV gene therapy delivers these protective dominant negative proteins directly where they are needed in the brain to prevent prion spread. Now, let me walk you through how we developed this therapy. We have two major challenges. First, optimizing AAV vectors for brain-wide expression. And second, finding the best dominant negative candidates. For the delivery system, we tested multiple AAV constructs with the different regulatory sequences. We evaluated proteins levels 3 weeks after administration. While all constructs expressed the dominant negative proteins, some construction clearly showed superior expression levels, and these were selected for our subsequent studies and confirm widespread expression across all brain regions. As you can see here, we achieved expression throughout the brain from the cortex and hippocampus to the cerebellum and spinal cord, as you can see here. Here, and also by immunohistochemistry. The results were excellent. We achieved optimal expression levels comparable to natural proteins levels with a stable, long-lasting expression throughout the brain, and importantly, safe delivery, no observed toxicity. Now, for the second challenge, finding the best dominant negative candidates, we had to select the most promising variant that could block prion propagation. This means proteins fitting with the first criterion, those that are resistant to misfold under laboratory conditions. And for this, we use what we call a NOAA-ARC approach. We analyze 944 different species from around the world, tested 424 different proteins variant for misfolding resistance, and use advanced laboratory techniques to evaluate each candidate. This comprehensive screening was like collecting samples from NOAA-ARC We wanted to find nature's own solution to prion resistance. Our second criterion was to select variants that interfere with normal prion protein conversion. We confirmed resistance to spontaneous misfolding and performed a series of in vitro studies. These included seeded propagation studies and seeded interference assays, which enable us to select the best dominant negative mutants using protein misfolding shaking amplification. Our third selection criterion was equally strict. We needed candidates suitable for brain delivery via gene therapy. This required using advanced technique, including NMR, spectroscopy, and computational modeling to characterize structurally our final candidates. And in order to obtain these structurally selected proteins. This approach identifies several highly effective dominant negative proteins for therapeutic testing. Now comes the exciting part, the results. We tested our therapy in two different disease models. First, we used the RML model in wild-type mice. In this model, we administer the gene therapy on the same day as prion inoculation, then monitor the animals until clinical signs appear for a human endpoint and sample collection. For this method, we tested one different dominant negative PRP. This slide shows the survival curve of wild-type mice inoculated with RML without any AAV treatment. This is the typical incubation time. When we administer the AAV expressing our dominant negative, the animals show a 27% survival extension. Importantly, the two animals that had incubation time similar to controls were not expressed the dominant negative PRP, which confirms the specificity of our therapy. Our second model was the TG-VOL, a transgenic mouse expressing BAM-VOL PRP. In this model, we first inoculated the mice with the CWD-VOL strain, which is the fastest acting prion strain known, and then administer the AAV therapy 15 days later. As before, animals were monitored until clinical signs appear for human endpoint and sample connection. In this model, we tested 4 distinct dominant negative PRP variants. This slide shows the survival curves of this model inoculated with CWD-VOL without any AAV treatment, 74 days. When we administer the AAV expressing for our first dominant negative, the animals show 21% of survival extension. When we administer the second one, the animals show a 22 survival extension. When we administer the AAV expressing our third dominant negative, the animals show a remarkable 44% survival extension. And when we administer the AAV expressing for our 4 dominant negative, the animals are still alive, as this is an ongoing study, very recent, but they have already shown more than 51% survival extension, which is truly remarkable. Let me summarize what have accomplished. We successfully identified highly effective dominant negative proteins through a screening of over 900 variants from different species worldwide. We achieved remarkable survival extensions of 21 to 51 in rapid prion disease model. We demonstrated both safety and broad brain expression of our therapeutic proteins, and we validated our approach in multiple prion strains. The impact of this work is significant. This represents the first successful gene therapy showing significant survival extension in prion disease based on this dominant negative therapy, providing hope for future treatment for CJD families. Our next steps include optimizing dosing for maximum safety and efficacy, testing in human prion disease model, CJD, FFI, and GSS, and advancing towards clinical trials. This research was made possible by the generous support of CJD Foundation families. As is indicated in this slide, your support makes this life-saving research possible and brings hope to families affected by prion disease worldwide. I want also to thanks to my people, collaborators, and people from my laboratory. Thank you very much.